Uh, Other things that happen to you, you might be in a competitive situation, you're going to be in a race, and your body has to immediately come up to speed for the race. You're faced with a threat. Uh, you pass somebody and you get a whiff of hormones, and maybe you're going to react to that. What this is all about is survival in the short term at the individual level. The changes are immediate, they don't last a real long time. I got to make that disappear. Durations like minutes, seconds, hours, depending. If you're too cold for too long, you're just going to die from it, right? And the effects are temporary. Once you go inside and you get warm again, all of that stuff goes away. That was a homeostatic reaction. Neuroplasticity is a little different. This starts out when you're born. When you're born, you've got lots of neurons, lots of dendrites, and they're all touching each other. And your brain is totally disorganized. And as you start to develop, you go through a period of dendrite pruning where your brain organizes itself to the reality that you're in. Then you start using your mirror neurons to learn by watching. Then you go through a period of developing muscle memory for all the things you have to be able to do, like walk. So for the first year of your life, you don't know how to walk. You develop a practice, you develop muscle memory, you record all those reactions in your cerebellum, and it becomes a reflex. And then from then on, you know how to walk for the rest of your life. And you don't forget how to walk. And also consciousness that lives in your precuneus. Then these are lifetime survival skills. And they not only apply to you, but they apply to your relatives. And if you're in a tribe, they apply to your tribe as well, because the more you know about, say, hunting, the better your tribe is going to do. The changes are lifelong. They last for hours, months, years, and the effects are lifelong. It's just like riding a bike, right? You've heard that before. Natural selection is multi-layered. There's individual selection where you choose a mate and you pass on your genes. There's sexual selection where you select a mate based on the quality of their genetics. There's kin selection where you help the survival of your genetics by helping your close relatives survive because their genetics are very much like yours. And there's something called group selection, which isn't necessarily what it sounds like, and I'm not going to talk about that today at all. But anyway, this is about survival at the species level. The changes are evolutionary. The duration is years, millennia. This is as close to permanent as you can possibly get, and it's multi-generational. So if you look at all of these, you see we've got a mechanism for different sets of durations: seconds, minutes, hours, days, months, years, millennia, millions of years. And then there's this gap, and that's where epigenetics fits in. This is decades, generations, and centuries. Epigenetics is the set of instructions in our DNA that tells our bodies, or it's the instructions on how to build an organism, how to build all the parts of the organism, how to set all of the settings in the organism for the current environment. So when you enter puberty, there's a time when you want to enter puberty. How does your body know when it's that time to go into puberty? And how does your body have to set itself up for puberty? And what hormones have to be generated? What proteins have to be generated? How much of each of these proteins have to be generated? How long is puberty going to last? And so on. It also deals with things like, how do you prepare for winter? If you're a bear, it's much different than if you're a frog, for example. Epigenetic changes are about survival at the seasonal level, at the episodic level, and by episodic, I mean episodes in your life. If you're a caterpillar, that's an episode, and then you're a moth or a butterfly, that's a totally different episode. And it's also generational, which is the most intriguing thing about this. Now, we haven't known about a lot of science for very long. I mean, DNA was only discovered in 1944. And this is the paper that announced the discovery of DNA. And you know, transformation of pneumococcal types. That's where we were. But once we discovered genetics, it was the firmly held scientific belief at that point in time that DNA drove everything, right? Your DNA determined who you were, and who you were was determined by your genetics. It was fixed, it couldn't change. Now I'm going to show you a little video. It's short, and it's jam-packed with stuff. But you know, bear with me, sit through it. Try to understand it as well as you can. If you have any questions, you know, raise your hand and we can address them. Yeah, how do you get out of here? Oh, there we go. Okay, so to recap, your genes are read by RNA polymerase and turned into messenger RNA. The messenger RNA is read by this machine called a ribosome, and for every 3 letters, it pulls an amino acid out of its hat, and it sets up these amino acids into a chain, and when you get to the end of the chain, it folds up into a protein. Now, why is that important? The reason it's important is because proteins do everything. This, by the way, is referred to as the central dogma of molecular biology. And I know dogma is generally a bad word in science, but there it is. But the thing is, proteins and non-coding RNAs do almost everything. So they are the interface to your body. If you have to hold oxygen in a red blood cell, you need a protein that's specifically designed to do that. And that's called hemoglobin. Salivary amylase is used to digest sugars starting in your mouth. Insulin regulates the metabolism of sugars, fats, and other proteins. Actin and myosin are used to make your muscles work. So if you want to flex a muscle, you need a protein that lets you do that. If you want to learn something, you need a particular NMDA receptor, which is a protein. So proteins do it all. 3 DNA bases make an amino acid. A gene reads all of these bases, creates a chain of amino acids. They fold into a protein, and those proteins do almost everything. So here's the question then. How does the RNA polymerase know when it's time to go read a gene? How does it know where to start? How does it know when to stop? How does it know which gene to read at all? And how does it know how much of a protein has to be produced? Because how much testosterone you produce depends on how old you are and what episode in your life you're going through. There are about 21,000 protein coding genes, and that seems like an awful lot, but it's the same number of protein coding genes that worms have and flies have and fish have. Now, 20% of those are used for coding proteins. Of your 3 billion base pairs, only 20% are doing that. So what is the other 90% or 80% doing? Well, 90% of your genes are transcribed. So 70% of our genes, we didn't know what they did for a long, long time. Not until the 1970s did anybody suspect that they did anything at all. And they were actually called junk DNA for a long period of time, which I found quite baffling. Now we know, or we estimate, that 20 to 40%, and that's a pretty broad range, right? But we estimate that somewhere 20 to 40% of that is for gene regulation. And what does that mean? Well, that's where epigenetics actually comes into play. You can think of the 20% that code for proteins as a hardware interface. If you were a computer scientist, then you would say, okay, somehow I've got a hardware interface into whatever device my software is running on. So if it's a phone, I have to talk to the hardware interface for the phone. If it's a computer, I got to talk to that, right? And if it's one manufacturer of computer, their hardware interface might be different than others. Well, for humans, the hardware interface to our bodies is these proteins, and that's what controls what your body is going to do, basically. And you could think of that regulatory stuff then as the software itself that's going to run and generate the information that goes down to the hardware. So this is where epigenetics is going to tell us how to do all the things that our bodies have to do and when to do them and how much and so on. And then the other 30 to 60%, we still don't know what that does. There's a guy, George Church, who works in a university in New York. He's actually built a microorganism and generated the DNA. It's got under 500 genes. It replicates, and he doesn't know what 2/3 of the genes do. Before cells differentiate, right, you start out as an egg, and the egg gets fertilized, and then it starts to divide. And in the early stages, these cells are not designated for any purpose specifically. So they're called embryonic stem cells, and they have to become something else. They have to become brain cells, blood cells, heart cells, muscle cells. So something has to direct what kind of cells they become. And they go through various stages. They start out as totipotent, and they can become anything, right? They can become skin, blood, neuron, whatever, pancreatic. And let's say, for the sake of discussion at this point, that there are a whole bunch of switches and dials inside the cell. And initially, all these switches and dials are set at the standby position. And you notice up at the top here, here's an egg becoming fertilized. Here it is divided into two, and it goes through a very early stage where it's called a morula, which is Italian for mulberry, because that's what it looks like. After that, it goes to the blastula stage, where it's got a lot of cells, and then it goes into the pluripotent phase, where it could become one of 3 types of cell: endodermal, where it could become a lung cell or a liver cell or a gut cell, ectodermal, skin, neurons, and brain cells, or mesodermal, skeletal, blood, and circulatory. So it starts off where all of those switches and dials are set to off. But if we select one of these things, you have to set the dials and switches for what it's going to be. And you can see here, as we go through these various phases, we get to this point where some of the cells, the ones on the outside, become placental cells. And the ones on the inside, they're going to become fetal cells. So that's the first real differentiation, right? These are the ones that are going to attach to the mother. And these are the ones that are going to become the animal itself. Then we go into the blastocyst stage. And in the blastocyst stage, some of them might become endoderm cells, and some of them stay pluripotent. And then they start to differentiate even further. And some cells differentiate early on, like your central nervous system cells, they start differentiating right in week 3. So the switches and dials for central nervous system cells, they get set pretty much first, then the heart, then the eyes, and then way out here, teeth and palate, and external genitals don't show up until week 7. So there's a lot of timing associated with this stuff too. So if a pluripotent cell, say this one here, is going to become mesodermal, the switches for that cell that say mesodermal are going to be switched to on, and all the others for endodermal and ectodermal, they're going to be switched to off. Then we go into the multipotent stage. So what's it going to be? If it's mesodermal, it could then go on to become skeletal or bone or fat or arteries and veins or part of the heart or white blood cells or platelets. So then it gets differentiated, and we've got some other switches and dials that get set to on, and then some of them get set to off because they're not going to become those things. So we've set switches for mesodermal, endothelial, and now we're down to it's going to be either an artery or a vein. So we switch the artery switch to on and the vein switch to off, and now that cell is an artery cell. Then it goes into the unipotent stage. It's fully committed, it's self-replicating, and when it replicates, all those switches and dials that said it was an artery cell have to say the daughter cells are artery cells as well, right? It can't accidentally become something else, because if an artery cell, when it divides, spits out a brain cell, that's not going to work in an artery. And it might even cause a problem. So you could say inside a cell, you've got this big machine, this big control center, and it's got all these switches and dials, and you got to turn some on and some off, and which ones, and the dials, what intensity do you set the dials at, right? And how long do you generate a particular protein? Does it last a month? Does it last a week? Is it for an hour? When do you turn it on? And you have to know what time it is, in effect, right? When is now? Is it time to go into puberty? Is it time to menstruate? Is it time to generate eggs and sperm? And then when the cells divide, all of these things have to be copied. And every time they divide, they all have to be copied exactly correctly. Otherwise, things will go wrong. But there are no switches. There are no dials. There are no timers. There are no sensors. There are no operators in there turning all these dials and throwing these switches. There's only DNA. And the DNA is made up of genes, and 20% of those genes generate proteins, and the proteins do everything, right? So if you generate a stem cell, some of these genes are going to be turned on, but they're going to be turned way down. Some are going to be turned way up. Maybe if it's a red blood cell, this gene's going to be turned off because there are aspects of a stem cell that are not necessary in a red blood cell, but they're really necessary in a neuron. If things go wrong, you end up with situations like this. This is a fruit fly, and this is a common problem that fruit flies have. Sometimes their antennae are miscoded and they turn into legs. And this is a fruit fly with legs growing where its antenna should be. So this is a case where Somehow those switches and dials did not get copied properly. Now you saw earlier what a chromosome looks like, and this is not what DNA looks like, right? This is all packaged up. And the reason it's packaged up the way it is into this tight bundle is because if you stretched out your DNA, it would be 2 meters long. And an exact copy of all of your DNA is in every cell in your body. So, and there are trillions of cells in your body. So if you can imagine stretching out all that DNA in every cell in your body, it would be a huge mass of stuff. So it has to be tightly compressed into this bundle. And if we were to look at that bundle, if we were to go to a section of that bundle, what we would see is it's all coiled up in these loops. And these loops, if we were to look at them, if we were to zoom in on that loop, you would see that there's a chromosome scaffold, and the loops are attached to that scaffold, and they're actually separated by this attachment. One of the things that can go wrong in life is this could break loose. And what happens inside this loop is supposed to stay in that loop. But if that broke loose, what happens in this loop might affect what's in this loop, right? Because there's no longer that barrier there. Now, if you zoom in on that, what you see is this thing here, it's called chromatin. And if you open that up, what it is strings of DNA. It's the string of your DNA, and it's one continuous string in a chromosome, and it's wrapped around this protein here, this set of proteins. And in between these proteins, with this 1.5 wrap, that's where the DNA is that gets coded into proteins. Now, these balls here, these beads on a string, these are called histones. There are 4 of them. They're different. And then they're replicated. So there's 8 histones in its 2 sets of 4 histones. When it's differentiated out as one set of histones with the DNA wrapped around it, it's called a nucleosome. There are 30 million of these in a cell, 30 million of these. And then in between them is the DNA that gets transcribed into proteins. Okay, so that's how it's all built. This is a microscopic, well, I think it's a recreation actually of a photo, a microphotograph of histones. So here's the DNA wrapped around the histones, and then the 4 histones are color coded. There's the blue one, and remember it's replicated. So here's the other blue one, right? The identical histone. Here's the green one and the green one, the yellow one and the yellow one back here, and then the 2 red ones. And these histones have tails. Each histone has a tail coming out. And that tail, that's where the dials and the switches are. That's where genes get activated or deactivated, or their expression is regulated and controlled. The histone tails are packaged in all DNA from yeast to humans. So this invention is one of the oldest inventions of life. Everything that's alive that has DNA works like this. Now, the expression of genes is controlled by proteins that are brought in by enzymes and attached to a tail, and that's called tagging. If an enzyme brings in a tag and attaches it to a tail, that's called writing. So there are enzymes that are called writers. And an enzyme can also come along and detach that tag, and that's called erasing. So there are enzymes called erasers. So some things that have to happen in your body, say you drink a lot and you have to produce a lot of the protein that's necessary to process alcohol, a writer will come in and it'll put a tag on that activates the part of your DNA that generates the protein that processes the alcohol. And when your alcohol levels get low enough, another eraser will come in, detach the protein, the tag, and turn off the generation of the protein that processes the alcohol. So it's an on-off kind of thing, and it's a temporary kind of thing. Some things happen episodically. All mammals have to become at some point lactose intolerant, with the exception of humans, oddly. But the reason they have to become lactose intolerant is so they'll stop nursing. Otherwise, they'd never stop, right? So at some point in a mouse's life, it has to become lactose intolerant, which means some enzyme has to come along and attach a tag that says you can no longer produce the protein that allows you to digest milk. And some things have to happen permanently. If you make a brain cell, it has to stay a brain cell and never turn into anything else. And if it divides, it has to make daughter brain cells and not anything else. So that would be a permanent modification to a histone tail. There are 30 million points along the DNA where tags can be added by writers, read by readers, removed by erasers. And if we were to show you an example of a histone tail as a chemical string, here it is. This is histone 3. And you see there are points where there are Ks. Those are points where a tag can be added to this histone tail. And that tag, you can have one tag or two tags. It can be a multiple thing. Some of these tags can be promoters. In other words, if I attach a tag at this point, which is K4 on histone 3, that promotes the production of a particular protein. And maybe I can add another tag, ME1, and that's an enhancer. You want to produce a lot of the protein, and so on. Each histone tail has multiple sites for modification through protein enzyme attachment or detachment. The Ks chemically are lysine. This attachment is called a methylation. This is a dimethylation because there's two of them. There are lots of points on the histone tails. Here are all of the different histone tails, and they all have unique tail structures. There are different kinds of chemical tags that can be added and deleted and so on. Okay? Now, before 1972, we didn't know this happened at all. There was a big mystery because we would look at things like variations in siblings, and we couldn't explain the differences in identical twins, and they called this intangible variation. An example of this is the caterpillar and the butterfly. They've got the same DNA in every cell, and yet they look completely different. And it happens episodically, right? It's a sudden thing. So we didn't understand what it was. They just gave it a name. Oh, that's an intangible variation. And that explains everything, right? And then we noticed some other crazy things, like the gender of a crocodile and certain amphibians is totally determined by how hot or cold it was when the eggs were developing. How does that work? And here's a queen bee. Her DNA is the same as every other bee in the hive. Same DNA, but she lives 20 times longer, and she's got wings, and she lays eggs. And the big difference is, well, she's fed a special diet. Now, what does this tell you? It says, well, if you're a butterfly, the period of your life is going to have a major impact on what's going on in the reading and writing of epigenetic tags in your DNA. If you're a crocodile, the temperature is going to have an effect. This is an external effect. And if you're a bee, your diet is going to have a major effect on what happens with your epigenetics. And now we recognize all these intangible variations as epigenetic phenomena. And it wasn't actually formalized until early 1990s. That's how new this is. So it's the epigenetic mechanism that triggers the expression of one or more specific genes at different times in your life. During blastocyst development, what's the cell going to become? During puberty, are you a male or a female? Are you going to produce this hormone or that hormone? And for how long and how much? Or are you going to fertilize an egg? Or you're going to become pregnant? All of this stuff is going to be controlled by epigenomics. And this story starts in the 70s with a mouse called the agouti mouse. And this mouse was bred specifically for the study of metabolic diseases. And one of the things about the agouti mouse is sometimes they're really fat and they're yellow, and sometimes they're small and they're brown, but they're still agouti mice. And when they're fat and they're yellow, they're prone to metabolic diseases and cancers. And so scientists use these mice to try and find cures for cancers and diabetes and things like that. They're genetically identical, and yet they're very different. And before the 70s, nobody understood why or how this could even happen. And the way it works is, here's histones. We talked about histones. Here's the DNA wrapped around the histones. And these little red dots in here, these are the tags. And on the smaller mouse, the brown mouse that doesn't get sick, the one that's healthy, on the smaller mouse, a methyl tag, a methyl group made up of hydrogen and oxygen, has affixed to a tail on the gene, and it shuts that gene down, and it inhibits the gene, and the gene does not produce the protein that causes that mouse to gain weight. And this methylation affects the color of the mouse, the obesity of the mouse, its vulnerability to these metabolic diseases. Another thing tags can do, and this is not methylation, this is called acetylation. Acetylation can cause these histones to be packed really, really tight. Now, if you remember in the video, RNA polymerase comes along and it reads the DNA between the histones, right? Well, if they're packed really tight, that RNA polymerase is too big to get inside and read the DNA. So that effectively shuts the gene off. And if you can tighten or loosen these coils, you can control whether a protein gets produced or not. What this is a picture of is the X chromosomes in the cell of a female. Now, a female has 2 X chromosomes, but they can't both be turned on at the same time because that would cause problems, right? You'd have 2 chromosomes trying to do the same thing with different information. So one of them has to be turned off. This one is the one that's turned off, and you can see it's been turned off using acetylation. It's packed too tight for the polymerase. I'm sorry, I'm getting tongue-tied. It's packed too tight for the RNA polymerase to get inside and read it, where the other one is opened up. And you can see there's lots of space between the histones where the polymerase can get in and read the DNA that's in between the histones. So genetic control via epigenetics is the normal mechanism that starts and stops all of the developmental processes that take place in a cell. It differentiates the 200 different cell types of the 70 trillion cells that make up a human being, and it enables responding to the environment faster than evolution allows. And all of these tags that control this stuff, that's what is called the epigenome. So you have your genome, your DNA, and you have your epigenome, which is all the tags that are attached to your DNA. And those tags come and go, and they change intensity and so on during the progress of your life. So what impacts the epigenome? Well, obviously where a cell is in the organism, right? Because a brain cell has to be a brain cell if it's in your brain, and where the organism is in its development. But there are other things too. Epigenetic marks can be influenced by the environment. So if your diet is low in folic acid, that's going to have an impact on your epigenetics. If you smoke, if you're subjected to a lot of stress, if you're subjected to harsh chemicals, these can all impact your epigenetic markers as early as before you're born. Now, they started to figure this stuff out in the 1980s. Scientists studied the birth and death records of an area in 19th century Sweden called Norrbotten. And people that lived there during times of famine, it was discovered, lived 6 years longer than those that lived in times of plenty. And the way they discovered this was in Sweden and in all the Norwegian or the Scandinavian countries, they keep really, really good records. And they had these records and they went through them and they saw that people that lived through times of famine lived longer, but they were overweight. They were susceptible to metabolic diseases like diabetes, and they had mental health problems. Now, it kind of makes sense that if you're going to live through a time of famine, you would want your body to store up as much fat as possible, right? And that kind of explains the overweight part. But what about the metabolic disease? Well, there must be some conflicts that if you're going to modify the epigenetics to force you to gain weight, maybe that's also going to force you to process sugar differently and cause the diabetes that also results. And maybe that's going to have an impact on your brain development and affect your mental health. But the really amazing thing was when they looked at these records, their children and the grandchildren of the people that lived through this famine period were also affected in exactly the same ways. Another example, women who were pregnant during a 1944 famine in the Netherlands known as the Dutch hunger winter had children and grandchildren who were unusually small and prone to diabetes and obesity. Same thing. 1998, Quebec suffered a major ice storm. Power was lost in some areas for months at a time, and the children of pregnant mothers from this time were followed for several years. They developed autism, more than average. They developed metabolic diseases, much more than average. They developed autoimmune diseases, much more than average. So nutrition obviously can impact your epigenome, if it's your epigenome that's at the root of all this. Is somebody trying to say something? I'm hearing a lot of hissing. Please mute your microphone. Now, what happened was, quite by accident, a female agouti mouse was fed the wrong diet. And when she was fed this wrong diet, it turned out that she didn't have the expected type of offspring. So I don't know what kind of agouti mouse was, but let's say for the sake of argument, it was the fat yellow one, right? She was fed the wrong diet. And instead of having pups that were fat and yellow, some of them were small and brown. So they looked at what they had done that might have affected this. They saw it was the diet. They analyzed the diet, and it was rich in methyl groups. And methyl groups are what form the tags for methylation. And those tags turn the genes on and off. And that's why she had the thin brown pups. And then those pups had thin brown pups. So, whoa, big clue. Now, when cells divide, epigenetic markers have to be copied by readers and writers. So if a liver cell divides, the readers have to read the tags and write those tags in the daughter cells so that they maintain their status as liver cells committed to being liver cells. There's a time, though, when this doesn't happen. If you produce sperm, that's a zygote. If you produce an egg, that's a zygote. When they get joined together, they create a gamete, a fertilized egg. And the markers, those tags, may not be cleared back to their standby condition, depending on what they are. When zygotes create a gamete, some of them have to be cleared a lot, and some of them cleared not so much. And it turns out that the tags from the father's side are scrubbed very aggressively, but from the mother's side, not as aggressively. So some of the tags that were set up in your body, if you're a male, are going to be reset. And if you're a female, maybe they will, maybe they won't. There are also imprinted genes, and imprinted genes are really interesting. If you get an imprinted gene from your father, it has a different effect than if you get that same imprinted gene from your mother. So those can't be scrubbed at all, and they're protected from scrubbing. Researchers gave pregnant mice chow containing only half of the calories they needed during the last week of gestation. What they're doing here is they're replicating a famine situation. And this is a time when the epigenetic patterns in a male embryo's primordial sperm are erased and then reset. And this treatment resulted in offspring and grandchildren that were underweight and prone to diabetes. So how does that whole thing work where it gets passed on? Well, here's an example. This woman's name is Sonia Hodis, and she married this fellow, I forget his name, Meyer Michelson, thanks, Lord. And Meyer and Sonia, they got married. Sonia got pregnant with a girl in 1915. And there's the fetus. And the girl fetus, they're going to name her Dinah later. At the time that fetus was formed, it included all the eggs that Dinah would ever carry. So Dinah's eggs, while she was inside of Sonia, were affected by Sonia's environment. So, you know, how good was Sonia's diet? Did she smoke? Was she subjected to secondhand smoke because Meyer was a smoker? Was she under a lot of stress? Then Dinah gets born. She grows up and she gets pregnant in 1948 from one of the eggs that was formed back in 1915. And now she's carrying a fetus that's 33 years old in egg years. And the egg and the fetus now are going to be further affected by Dinah and what she does and her environment. So if Dinah makes good choices about diet, vitamins, drugs, alcohol, tobacco, stress, and so on, did she live through a famine? Did she live through a war? This might change the tags that were created in the eggs that Sonia made way back then. And then Lori gets born in 1949. Her egg age at that time was 34 years old. The epigenome now includes the effects of her grandmother's life and her mother's life and lifestyle choices. When cells divide, they create a gamete, and the markers may or may not be cleared back to the normal state. And if they don't get cleared, they become an inherited epigenetic trait. So not only do you inherit genes from your parents, you inherit tags on those genes from your parents. The addition of environmental toxins to the diet of an agouti mouse can cause the epigenetic change to disappear. That's called demethylation. One of the ways this can happen, and they've done this experimentally, you got a fat, yellow female agouti mouse. If it has pups, they're going to be fat and yellow. If you've got a brown, skinny agouti mouse and it has pups, they're going to be brown and skinny. But if you feed them alcohol in significant quantities, they don't come out as expected. They come out mixed. The fat yellow one has some that are small and brown. The small and brown one has some that are fat and yellow. Another experiment was done with fruit flies, where 2 days of dietary sugar in large amounts were given to the male fruit flies, and it elicited obesity in the offspring. There are different ways things get turned on and off. There's methylation. That turns a gene off and on. There's acetylation. That's the one that tightens and loosens the nucleosomes. There's microRNA. We didn't talk about this at all, but remember when we were creating the messenger RNA, and that was going to be used by a ribosome to produce a protein? A microRNA can come in and attach and block it. And that blocks the transcription of the protein, so the protein doesn't get produced. In addition to these mechanisms, there are 80 more that affect the modifications of histone tails. And the code to this, we don't understand very well at all. This is a really new science. It wasn't formalized until the early 90s. We're just now figuring out how it works, and whoever figures this out is going to win a Nobel Prize. Here are two identical twins. They're literally clones. Their DNA is exactly the same. They have identical genes. When they're very young, they act the same, they talk the same, they have the same body language, they have the same potential abilities, but one of them might get cancer and the other might not. One of them might be schizophrenic, and then the other one would only have a 50% chance of being schizophrenic too. In addition to epigenomic changes, identical twins may accumulate somatic DNA mutations as they age that make their DNA not completely identical. But there are many diseases that we suspect are impacted or caused by epigenetic dysregulation. Now, what this is a segment of DNA that's been overlaid from two identical twins. And where their DNA is identical in tagging, it's yellow. So very, very much the same. Two identical twins, both of their DNA overlaid, and their epigenome is almost identical. Here it is, same thing, but their DNA is totally different in tagging, because you don't see much yellow at all. You see a lot of red and a lot of green. Well, that's where the epigenetic tags are different. And what's the difference? This is when they were 6 years old. This is when they were 66 years old. So over a 60-year period, they experienced different lifestyles and different exposure to food and chemicals. And so their tagging is very different. So exposure to diet, exercise differences, chemicals, smoking, medications that they take, stress, these can all lead to the suppression or hyperexpression of proteins. Here's an example of two monkeys. They're from the same litter. This one is raised by a mother monkey, her mother, but this one was separated and was raised by a nurse. The one that was raised by a nurse grew up to be more aggressive, unable to deal with stress as well, and more prone to being an alcoholic. And here's a mapping of their epigenetics. And you can see their epigenetics based on high methylation is red, green methylation is low, right? They're very different in methylation. These monkeys are methylated in entirely different ways only because of whether they were raised by the mother or by a surrogate. So what happens is a mother rat will lick its offspring. And if it licks its offspring, that affects the amount of serotonin produced in the brains of the pups. And there's a gene called the glucocorticoid receptor. And that is used to handle stress. And if the pup is licked, or sorry, initially that's turned off, right? So if the mother licks it though, transcription of that gene is unblocked. But if there is no mother behavior, no licking and grooming, then the transcription of that gene is blocked. And because it's blocked, the pup grows up unable to deal with stress throughout its entire life. So having a lack of motherly attentions makes the pups unable to deal with stress, and it changes their sexual behavior. So here's a warning. If you're going to have kids, you better treat them like a good parent. Otherwise, you might be doing something to their epigenetics that's going to hurt them later on in life. Affection generates serotonin. The serotonin has an effect on a specific epigenetic tag that later impacts the pups as adults. Aristotle said famously, and it's been misattributed many times, give me a child until he is 7 and I will show you the man. What he's actually saying, though, is you can give a child love and affection, or you can ignore him. You can give a child nourishing food, or you can feed him junk food. You can give a child a safe, comfortable environment, or you can make his life a living nightmare. You can give him a clean environment, or you can expose him to secondhand smoke. And all of these things could have an impact down the line. Abused and neglected children tend to be alcoholics. They tend to be drug addicts. They tend to be suicidal. They have more mental illness, and they also tend to be more socially maladjusted than the average. So abuse and neglect are bad. So how much you're touched as an infant, how much you're encouraged as a child, how many hours of sunlight your mother was exposed to when she was pregnant, how much nourishment was available to her and to you, how clean the water was, how clean the air was, how often you were frightened, how badly and for how long, and how much exercise you and your mother both got in the early years. All of these things have an impact. So for a long time, the question was, is it nature or is it nurture? Well, consider this. Suppose you were born 20 years ago in Iraq. What would the impact of growing up in a war zone be on your weight gain, on your emotional quotient, your intelligence quotient, your empathetic abilities? What's that later going to do to your relationships and your ability to have relationships, and on your ability to be a good parent, and what you're going to pass on to your offspring, right? And these are things that would be totally out of your control just based on where you were born and where you grew up and what was going on around you at the time. So is it nature? If it's DNA, yeah, it's nature. If it's nature, it's DNA. Is it nurture? It is if it's your epigenome. And if it's your epigenome, then it's nurture. And it turns out that they are almost equal in impact on what happens to you later in life. Now, DNA, if you get a defect in DNA, we are just now learning how to attach or attack those DNA mistakes. But for the most part, your DNA is fixed. But if it's epigenetic, there might be something we can do with those tags. And if we can affect those tags, then we might be able to fix mistakes. We might be able to reverse them. So in DNA, if you get a mutation, that causes a disease, and it's called a monogenic disease because it's one gene that's causing it. And that's like cystic fibrosis, sickle cell anemia, muscular dystrophy. If it's a complex disease, it's probably polygenic. It occurs outside the genes, and these are diseases like diabetes, especially late onset diabetes, right? Schizophrenia, which happens later on in life, multiple sclerosis, which happens later on in life, cancers. They're influenced heavily by the environment, and they're very likely to be epigenetic. There's an experiment they've done with lab rats, and I feel bad for lab rats, I really do, but not so bad for this one, because he's become a cocaine addict. And he's got a mechanism here where he can step on this pedal and give himself a dose of cocaine. Well, this rat will give himself doses. He won't eat. He won't sleep until he passes out. He won't have sex. He'll dose himself until he dies. But increasing methylation increases the desire for the drug and decreasing methylation can cure the rat of its addiction after a single treatment. So drug addiction is an epigenetic, curable condition, at least in rats that live in laboratories. Now, not all diseases are caused by epigenetics, but those that are may be curable through epigenetic reprogramming. And I don't use the word reprogramming lightly. If you think of epigenetics as the programming that controls the hardware interface that is the DNA producing proteins that control your body, we're talking software here. In 2009, researchers at Duke University Medical Center published a study in the journal BMC Medicine on epigenetics and autism, and they found that some children, it was a small study, 40 kids, but some children with autism had extra DNA referees turning off a gene needed to respond to oxytocin. And oxytocin is the hormone, that's the drug that makes you feel good when you have social interaction. Now they use a phrase here, DNA referees. I'm going to talk about referees more in a minute. Until recently, scientists thought genetic mutations played a bigger role in cancer risk than epigenetic changes to our DNA, but lately it's become widely accepted that epigenetics may play an equal, if not a bigger role in cancer than genetic mutations do. There are tumor suppressor genes that protect cells from becoming cancerous, and epigenetic tags can turn those genes off. So if that happens, you would be more prone to getting cancer because you wouldn't have the suppressor genes there. So drugs that could target these tags to remove them would allow the tumor suppressor genes to perform their function. Here's an actual cure for lung cancer that was developed in 2006 using epigenetic information. Here you can see the tumors. This is in the lower lungs, the upper lung. And here's the lower lung. And you can see the tumors there. And this is after 8 weeks of treatment with epigenetics. And you can see the tumors are hollowed out. They're dying. So it works. And this was taken orally. This is basically a pill. The primary principle behind this therapy is you target the epigenetic changes with a drug that reverses the damage and returns the expression of the DNA back to its normal precancerous condition. So epigenetic drugs, or epidrugs, are approved by the US Food and Drug Administration to treat epigenetic-related positive metastatic breast cancer, cutaneous T-cell lymphoma, etc. Other candidates for epigenetic cures: rheumatoid arthritis, type 2 diabetes, addiction, asthma, lupus, obesity, Alzheimer's, Parkinson's, autism, schizophrenia, bipolar disorder, and Huntington's disease, and there are many others. And all of these diseases have something in common. They all happen later. You don't get these things when you're born. They all happen later, which indicates that they're probably epigenetic. And your epigenome, as we saw, changes over time. The tags come and go and accumulate, and things break down and so on. And if you could look at a map of your epigenome and you could see where it sits in this chart, you would determine what your epigenetic age is compared to your birth age. So if your epigenetic age is 80, but your birth age is 60, hey, that's bad, right? That would be a dot up here somewhere. But if your epigenetic age is 40, it's down here, and you're 60, that's good. And the median error here is just under 2 years. So this is a very accurate thing. And you could look at this and say, wow, I've been living a really good life. I'm up here above this line, or I've screwed up and I'm way down below this line. Some of the things they found by using this method is they found that children age epigenetically much faster than adults. And that makes sense, right? Because when you're very young, that's when you're going through all your rapid development stages. Men age faster than women. Now, this isn't necessarily a fact of being a man. It could be that the lifestyle you live as a man is different than the lifestyle that women tend to live. Maybe that's the impact. Aging signs are impacted by life exposure and lifestyle. Chronic stress accelerates aging. Chronic stress, but acute stress for short periods doesn't. And cancer cells age faster than normal tissue. So once they go off the rails, they continue to go off the rails. All of this stuff comes later in life. And it turns out that 2/3 of all deaths are attributed to chronic diseases that come up later in life. And 75% of healthcare cost is attributed to this kind of disease. So most of the stuff that's killing you and most of what we're spending our money on is epigenetic diseases. And they usually involve the dysregulation of normal gene expression. Cell types can forget what they are due to epigenetic changes or mutations to DNA. A broad opening of chromatin due to loss of histones can be a cause for that. A mutation amongst the enzymes that do the reading, writing, and erasing could be a cause of that. So you want to find an epigenetic cure for a disease. Well, it's a really complicated problem because you got to find the right drug, which means you got to know what the right chemical is for a particular tag on a particular histone, on a particular gene, somewhere in a particular cell type that's causing the expression of the protein or the repression of the protein that's resulting in the disease. Complicated stuff. The efficacy of the mitigation of aging-related issues, though, can be tested using that epigenetic clock I showed you. And there are people, HIV-positive people or people with Down syndrome, you look at their epigenetics and they're above the line. Their epigenetic age is much higher than their birth age. The general population falls on this line, and people who are the offspring of other people that live to be over 100, they're below that line. So if I gave you a drug that was supposed to make you younger, I could look at your epigenome and say, yeah, it works, or no, didn't work. And we could tune that drug and figure out if it's working, if it's not working, how can I make it better, and so on. Now, there are some things that you can do. So this should be really important to you now, and you should tell your children and your grandchildren. I mentioned referees before, DNA referees. One of the things that has to happen is suppose you get a cell that's told you're a brain cell, and the enzymes come along and they switch on, the brain cell switches and the dials for brain cells. And then that cell produces proteins, and those proteins kick off another response in another cell that produces another protein that feeds back and says, yep, you're a brain cell, you keep being a brain cell, and it goes round and around in a feedback loop. And that's how a cell continues to be a cell. That's called a DNA referee, that once a cell has been determined and committed, it stays that way. Other cells have to switch on and off in their functions, and it's a little more complicated. But again, it's a referee situation where you have a feedback loop that changes. Sometimes it's on, sometimes it's off. And all of this stuff is happening all the time, which means the chemicals that are necessary to do methylation and demethylation and acetylation and deacetylation and so on, all of those chemicals have to be there all the time. And you can see there's B6, B12, B2, methionine, betaine, choline. All of these chemicals have to be present in your body in sufficient quantities to constantly be doing the things they have to be doing. And if you don't have them, that's going to accelerate epigenetic aging. So things that impact the presence and the efficacy of those chemicals is going to hurt you. Vitamin E from soybean oil and corn oil is bad for you in terms of epigenetics. A lot of sugar in your blood is bad for you. High triglycerides, bad for you. High insulin, bad. High blood pressure, bad. High body mass index, bad. Processed foods are bad for you. Certain food colorings are shown to impact negatively your epigenome. Some pesticides are known neurotoxins, which adversely affect IQ scores, probably because they're impacting your epigenome. I mean, Roundup has been in and out of court many times. Air pollution is a grade 1 carcinogen. Well, if it's a carcinogen, that means it's hurting your epigenome. Scientists at Keck School of Medicine found out that if a mother smoked during pregnancy, there were long-lasting changes to the children's epigenome, usually around cancer and development, and over 50,000 genetic variations are possible results of smoking. Paper in 2009 in Nature Neuroscience, 12 people who were abused as children and later committed suicide had different DNA referees on a gene that was needed for coping with stress. So here's another set. These are called endocrine disrupting chemicals, EDCs, and they're tagged as obesogenes. And what do you think they do to you? They make you fat, right? Obesogens. And two of the most powerful ones are tributylin and dibutylin. They're used in the manufacture of hard plastics. So hard plastics, obesogens. Bisphenol A, this is another toxin common in hard plastic, affects the epigenome of unborn mice. But eating folic acid counteracted those negative effects. So that tells you that you can, with your diet, have a positive impact by replenishing the chemicals necessary for the proper tagging of your epigenome. So to slow epigenetic aging, if you want to stay young forever, a diet of poultry, fish, fruits, and vegetables, a lot of fish particularly, has the greatest impact. Carotenoids, carrots, sweet potatoes, and green leaves, like your grandmother used to tell you, eat your greens, honey. Xanthines, turns out, here's the good news, caffeine and chocolate are actually good for you. If you want to get vitamin E, get it from olive oil. So stop cooking with corn oil, start cooking with olive oil. Moderate alcohol assumption, that's my favorite, is actually good for you. Chemicals, folic acid. So if you're going to take vitamins, you probably want to make sure you're getting folic acid. Of course, you can eat a lot of broccoli and beans and get it that way. Garlic is another good source of chemical tags that are part of the epigenome. Brussels sprouts, kale, your B vitamins. B vitamins are very important. So that's basically it. And now you know what epigenetics is about, how it works, and why it's important. So I'm going to stop sharing now. Let's see. I don't know how to turn this off. Dan.
Thanks so much, Ron. That was
I don't know how to turn
information
this off.
packed.
Eric, can you turn this off? Oh, wait a minute. I found it. Found the button. Yeah, I know. It's a lot. It's a lot. And I cut about a third of my slides out because normally when I do this presentation, it takes almost 2 hours, and I wouldn't do that to you guys. So any questions, thoughts, comments? To me, this is one of the most important things you can know about, because I mean, this actually affects you and it affects your children, whether you know it or not, and affects your grandchildren, whether you know it or not. And you should tell your kids, especially if they're of childbearing age, about this stuff, because you know, people don't know. They just don't know. People do some pretty cavalier behaviors around like smoking and exposing themselves to secondhand smoke and the like, and it has an impact.
Yeah. So a lot of these things we understood at a very sort of intuitive level, and now epigenetic research is showing us the mechanisms.
Yeah.
And people are starting to play around with trying to give people compounds to affect this. The first attempts are going to affect it in fairly indiscriminate ways, I would imagine. So I wonder whether the early treatments to this might end up having a lot of odd side effects, because if you just suppress methylation generally or accelerate the methylation generally, you could have a host of very unpredictable effects that we really don't yet understand. So I would imagine the first treatments are going to be very risky.
Not necessarily. Like the cure for the lung cancer that we saw. If you identify the protein that's causing the lung cancer, right, or the suppression of the protein that would protect you from the lung cancer, and identify where that's coming from, right? What cells? Well, if it's in your lungs, it's probably your lung cells, maybe. So you would look there first, and you would look at the methylation patterns of those cells and compare them to the methylation patterns of people who are free of lung cancer and very young, right? And you would look for the differences there. And hopefully that would enable you to pinpoint what cell do I have to attack, and what is the methylation that's different, and how do I attack that specifically? And that's what you target, right? And the other thing you have to consider, yeah, go ahead.
Are there methods to be that targeted, to attack a particular tissue type and a particular methylation site on one of those tails?
Yeah.
Or really?
In fact, I gave this presentation out in virtual reality, and one of the guys who attended actually worked in a laboratory, and that's what he does for a living. Is he is the guy that's trying to identify those tags. And I talked to him at some length about it, and yeah, it's tedious, but they're doing it, and of course it's all being done with things like agouti mice before they ever get to a human being. So yeah, you might screw up a mouse or two before you move on to maybe monkeys, right? And so on. Leland, you got something?
Yeah. Several years ago, I sent a sample of my DNA to 23andMe, and they read my genome and they told me all sorts of wonderful things. Can we, or how close are we to being able to send in whatever and have them read my epigenome and tell me whether I was abused as a child or whatever?
Oh, there is a company that will read your epigenetic clock and tell you, your epigenetic age.
Yeah, but I want to know what tags are on and off.
I don't know if they'll tell you that. That's getting pretty deep, right? No. And they also have that epigenetic clock for various animals, by the way. You can have the epigenome of your dog studied, or if you have a horse. They've done it with a few animals, not a bunch, but 3 or 4.
But I assume it's theoretically possible, right? I mean, with one.
Yeah, absolutely.
Are the tags on cell 1 the same as the tags on cell 2 within me?
No, they're not, because it depends on what kind of cell it is and what the purpose of the tag is. Because if you have a brain cell that's tagged as a brain cell, those tags are going to be a lot different than a cell that's tagged as a lung cell, right? So 2 brain cells hopefully would be tagged the same, and if they weren't, then maybe you got the start of a brain tumor going.
Okay.
So differentiation in tags for identical cells would be a bad thing.
Ron and Eric, I'm sorry, I have a question.
Yeah,
I would
Chris?
like to share
Uh,
today's presentation with my daughter who is pregnant.
yeah.
How can I, is it okay to do that? How can I share the recording?
Oh, share the recording? I can give you a copy of my slides. I haven't. Eric, are you recording this? Oh, Eric says it's being recorded, so check with Eric. He'll
Thank you.
probably post it on YouTube, maybe.
Eventually.
Yeah, eventually. Eric's talking, but we can't hear him because he's muted. It's one of those dumb things we do.
Ron, can I have a question?
Yeah, Chris.
Yes. First, a little story. Okay. Some time ago, a Polish writer, a woman, she's quite popular. She came up with the idea. Actually, she lived in many countries, pretty much like myself, so we have a pretty good communication. So she came in public with the idea that a lot of problems of my country come from a culture of alcohol abuse for many, many years, actually for hundreds of years, which actually was proven even in literature. And obviously, a lot of people were so emotional about this, saying, okay, you leave this country, you don't take her passport. My question to you is there possibility, is there a probability of like whole society affected by the culture, which actually is like popularizing or maybe given kind of tolerance for the factors affecting epigenetics, right? Epigenetic process. Is it possible? Because
it's definitely happening. If you go back to Victorian era England, and well, the Victorian era in general, people were very fond at that time of green wallpaper. And if you were a rich person, you could afford to put up green wallpaper. Turned out the green wallpaper was made green by arsenic.
Ooh.
So if you were a wealthy person, you were living in a house and you were surrounded by arsenic. Another thing people did in the Victorian era was they would put their kids when they were very young in a kind of a cage. Children were meant to be seen and not heard, and they would actually be put in a cage so that they wouldn't disturb you when you were, being sociable. And what effect would that have on kids, right? Because instead of being treated with affection and developing serotonin to enhance their glucocorticoid receptor so that they would be able to deal with stress later on in life, they were being treated like that. If you're born poor, right, you're not getting the nutrition, you're not getting the vitamins, you're dealing with a lot of stress as a very young person, just from the fact that you're being poor. And that's going to have an effect on you. One of the biggest crimes we can commit as a society is allowing people to be poor, because it hurts the entire society. And alcoholism, absolutely. If in Russia, they take great pride in getting stinking drunk on a regular basis. Well, that's going to have an impact. It's going to have an impact on the epigenome. If you're doing it while you're pregnant, if you're doing it while you're producing sperm, it's going to have an impact on your offspring.
Thank you so much. Yeah, that was exactly when I was growing under communist Poland, just because of the society being so like depressed and so much strictly controlled. Alcoholism was a huge problem, pretty much like in Russia. That's what we're talking, cultural.
Yep.
So, well, there was a lot of hope after this change with this whole transition, that now there will be completely different country. And many thinkers now come to the point that we have the same problem, no matter we have
Yeah.
completely different system affecting in all ways. So, well, I tried myself to raise this issue a little bit, but so far there's a problem because they don't want to accept it. They're not ready for this yet. No matter,
Well,
there are some scientists already popularizing this, some young scientists, but still they are just very selective. They want to hear what they want to hear.
sure. And you know, religion has an impact too, because some religions are much more restrictive than others. Some religions promote being, you know, a loving parent, and others promote being a very severe parent. So, you know, that's going to affect the kid.
Very good.
Yeah.
I have a
Thank you
question.
for this. All right.
Yeah.
Is there, hello, is
Yeah.
there
Yeah.
a geographical, a racial component to this? You know, you talk about hours of sunshine, and if you live on a river or lake or near the equator.
Yeah, I would assume so. I haven't read anything specifically about that, but you know, given the importance of diet, there was one study I read about that took place in a part of Africa. Maybe the location will come to me, but it's a place where they have a seasonal drought, and children that are born during the drought period are different epigenetically from the children who are born when the rains come and there's a lot of green leafy vegetables to eat. So the parent, the mother, if she's pregnant at the late end of her pregnancy during the drought period, the kids are going to be impacted one way, where during the rainy season, she's impacted totally differently. And this is a seasonal thing, and it goes back and forth. So if you could pick when you were going to be pregnant, if you were smart enough to do that, right? If you were educated enough to do that, you could have some control over it. But if you're totally unaware that this is going to have an impact, then you think nothing of it, or you have no control over whether you get pregnant or not is another factor. So yeah, that's totally localized.
Can I say something about this, about your question? Because that's exactly our experience. In 2003, we went back to Poland. And first couple of years, that was absolutely disaster. I just couldn't just live over there. Later on, there was a plan to open branches, and we moved to Spain. And then everything, okay, got back to normal. So for all 10 years, being in Northern Europe, in UK, I spent like 3 months in Spain every year. And, you know, there was the same thing. So in April, I was drained. Okay, and then back in September, I was full of energy, which was going lower and lower. I spoke to the doctors. They were just looking at me like, you know, like coming from different planet. But later on, they found out that some of vitamin D is not produced in northern countries. I didn't know it. I lived with that for many years. But once I spent like almost 20 years here, at well, having much more sun, no matter it's not tropical country, I couldn't adjust to that northern climate anymore because I was older. So exactly, that was exactly the experience. Okay, we had that every year, going down in April, couldn't wait. Then summer, Spain, perfect, and then coming back again. Okay, we had that.
One thing about the vitamin D is that in the northern climates, you're correct. In the winter and fall, even if you stand out at 12 noon, you will not create vitamin D. However, you will create vitamin D during late spring, summer, into early fall in northern climates. But it's the winter, like now, where if you don't supplement with vitamin D3, and particularly if you're dark-skinned people, you're going to have a far lower level of vitamin D than lighter skinned people do. Also, I wanted to say one thing about crime and leaded gasoline.
Mm.
There was a period where crime went down in most of all cities, and they weren't really able to find out any logical reason why this happened. And I don't know if you know this, Ron, one of the hypotheses is when we changed from leaded gas to unleaded gas, that coincided with a lowering of crime.
Yeah, I read an interesting article one time about the fall of the Roman Empire, and one of the premises that was put forth is they did it to themselves because the rich ate off of lead plates and drank out of lead goblets to demonstrate how rich they were, and they all gave themselves lead poisoning and became really, really stupid. And that was the ruling class. So you've got that. You've got the Victorians living in arsenic. And today you've got plastics. The average American consumes a credit card's worth of plastic a week. And plastics impact obesity at the epigenetic level. So if you look around and you say, wow, why is everybody so fat when, 50 years ago, people weren't fat like this? Well, 50 years ago, you weren't eating a credit card a week worth of plastic. And maybe that's what's causing it, right? Patty?
There's also a story about folic acid. People talk about folic acid, and you do also, Ron, on your slides, but it might be better for you to say folate instead of folic acid, because folic acid is a synthetic molecule. And if we eat too much folic acid, it causes cancer. It's carcinogenic.
Wow. Well, I got a question for you.
Because wait,
look,
hold on. There's the thing that you should know is a significant percentage of people cannot metabolize this synthetic molecule. So it builds up in your intestine, and that's what the problem is. So these people get no benefit from taking folic acid in their vitamin pills. They need folate, which is the form that really goes into cells, and that what we can use. So nobody should be taking a vitamin that has folic acid in it. You should be taking pills that have folate, but not folic acid.
Right. I looked at my bottle of folic acid, and on the label it says folic acid, but on the list of ingredients it says folate. What should I believe?
I would say that it should say folate and never say folic acid. And actually, it's a form of folic acid that many formulations have. It's a 5-something-something methyl something folic acid, which is the exact form that goes into your cells. So yes, so if you just take folic acid in your vitamin pill and have a low folate diet and you're pregnant, then you're more likely to perhaps have a problem with your fetus.
Thank you. Patty's a real biologist. I only pretend to be one.
Hey, Ron, where is the principal source of this credit card amount of plastic? Is it in the packaging of food? Is it in the food itself?
Well, there's twice as much plastic in bottled water than there is in tap water. So, if you're drinking bottled water because you think it's a healthy thing to do, stop doing that. Unfortunately, it's in fish because we throw a lot of our plastic into the ocean. It gets ground up into microplastic and the fish swallow it and it becomes part of the fish. So very large fish have a lot of plastic in them, where small fish tend to have less. So one of the rules about eating fish is never eat a fish that's bigger than the plate. So, you know, you can do that, but plastic is everywhere. I mean, we're swimming in it. It's a really bad situation.
Ron, I tried to email you that YouTube thing about B vitamins and omega-3 fatty acids, and all I sent you was Chinese gibberish, I think.
Yeah, thank you
But
for the Chinese.
this is actually a very important thing that you can all watch on YouTube. And it's entitled How B Vitamins and Omega-3 Reverse Cognitive Decline. And it's done by the head of the pharmacology department of Oxford University. And I've been following omega-3 fatty acids, and maybe some of you have, in terms of Alzheimer's and cognitive decline. And it has had some success, some mixed results, some negative. Same thing with B vitamins. They have been pretty sure that B vitamins, particularly B12, folate, B6, help with cognitive decline. But what this latest study did was it went back to the earlier omega-3 studies, and it parsed out the people who had high levels of B vitamins. And those people with the omega-3s definitely showed an improvement in cognitive decline and an improvement in not getting Alzheimer's disease. So for the first time in the last 12 to 18 months, this is kind of new information. They found that for both of these to work, and this English pharmacologist said the drug companies would have spent billions of dollars, and they wish they could get halfway towards what the combining of B vitamins and omega-3s can accomplish, particularly as I look at all of us who are all over 25 years old, yeah, times 2 or times 3. So this is a very excellent YouTube, and it's How B Vitamins and Omega-3s Reverse Cognitive Decline. You go on YouTube, it's an hour and 20 minutes, well worth your time. I think you'll find the information informative, and maybe will change your approach to this problem.
Yeah, there's I forget the name of it, but there's one of those pills you can go to the drugstore and buy that's supposed to improve your memory. I should probably take. I can't remember the name of it.
I can't.
But anyway, I went to the drugstore to look into that, and it turns out a bottle of that stuff costs $50. It's got 50 pills in it. No, it's got 30 pills in it. Lasts a month. And on the label, it clearly says you have to take it for 3 months before you can even hope to see an effect. So you got to spend $150, and maybe it's going to have an effect you can notice, you know, where, as you say, you know, take your vitamins, eat the right food. That's what's gonna work.
And B12, particularly for my daughter who's a vegan, is particularly important because you don't get B12 if you're not eating animal foods.
Yeah.
And even if you take a multivitamin B12, B12 is one of the worst vitamins to assimilate into your cells. And what I do is I take from Kirkland, Costco, they have 5,000 IU that go under your tongue. And I do that about twice, sometimes 3 times a week, if I remember. And I take a combination of fish oil and algae, and I recommend it.
Yeah, Prevagen was the thing I was trying to think of.
Prevagen, yeah, that's the big one.
Yeah, waste of money. Oh, another interesting commercial I've seen lately is from Wegmans. Maybe you've seen this. It's the modern family. They're all running off to work and to school and everything. And the mother has a plastic bag of sauce that she throws into a slow cooker in the bag. And it cooks all day in the plastic bag. And then they come home and have pasta. So you wonder where the plastic's coming from. I don't know if I'd do that one.
Art, are you able to put the links to the thing to that video you want us to see in the chat?
I'm not good at that,
or something we can Google?
Just go onto YouTube and type in how B vitamins and omega-3s reverse cognitive decline. And you'll find it. And if the guy has an English accent, and if he's the head
well, that makes him more believable, right?
Yeah, oh, we all believe people with English accents. But he has some terrific research on, I thought, very, very moderate, very scientific. So how B vitamins and omega-3s reverse cognitive decline. Google that, or go onto YouTube and type it in, and that should be the one that comes up.
I'm trying to put that in the chat myself.
Leland beat you to it.
Okay. Oh, he actually has the link. Okay, cool.
I wanted to make a comment about fish. The smaller fish, sardines, for example, have less mercury, and the bigger fish, like the tuna, have more mercury. So maybe that's also true with microplastics. The smaller fish are better because they don't live long enough to accumulate. They accumulate less mercury as well as maybe microplastics.
Well, don't forget too that fish eat fish. So the little fish get eaten by bigger fish. So that's how they're accumulating all of this stuff too, just by their daily diet. And when a fish gets as big as, say, a tuna, it's eaten a lot of mercury and plastic.
Yeah, well, one of my friends got mercury poisoning from eating too much sushi. So that was a lesson too. The other thing about the water is I recently stopped drinking tap water. I was filtering it with 2 different filters, but Environmental Working Group made a statement about swimming river reservoir, that there's radium in there, and any radium is too much. Now, the radium in swimming river is less than EPA levels to be harmful, but Environmental Working Group says any amount of radium is detrimental to the body. So I had switched to bottled water recently now, and it does taste plasticky. So I'm using one of my filter, the zero water filter, to filter it out, but I don't know if it filters the microplastics in the water. Probably doesn't.
Don't know.
If you go on Consumer Reports, which I did, I never had an under sink filter before. I do now, and it cost $100. I had to have someone come in to put it in, which was another $150. But they were doing plumbing anyway, so that's why I went with this under the sink device. It's not reverse osmosis. It's which is expensive and can be problematic. It's a different kind. I'll let you guys know what the name of it is. And I've had it for 6 months, and I use it just for drinking. I don't care what I shower with or do dishes with. So it's solely for that. And that has solved my problem of what water to drink. I also make ice cubes with it.
How do you know when to change it?
It comes up red when the filter is no longer filtering properly. Let me see if I can find the name. You want to hold on. If I can see.
He's crawling under the sink as we speak.
Right.
Don't hit your head.
So getting back to some of the epigenetic changes, there's definitely been a switch towards being more nurturing of children and protecting children from various exposures in the last even 50 years relative to the early part of the 20th century where people had a more spare the rod, spoil the child attitude. Can we see any measurable effects of that are positive in the same way that removing lead from gasoline may have been positive?
Well, one of the interesting things I took out of the presentation was a slide that talked about how people that are wealthy have better epigenetics than people that are poor, because that's not a direct link. What it does tell you, though, is if you can afford it, you can afford to live well, you can afford to live without stress. If you can afford to live in an environment where people are educated and loving to each other, then of course you're going to be better off epigenetically. What I think the message there, though, is, what if you can't? 40% of Americans are $400 away from bankruptcy, right? Which means, a set of tires can put you in a really precarious position. Well, people that live like that are probably epigenetically in a very bad way because they live with a lot of stress. Their grocery bills. I mean, when I first got married, Lori and I would spend $25 a week on groceries, $100 a month, and we ate whatever we wanted, right? $100 a month. Now, our grocery bill is over $1,000 a month, and we don't eat red meat anymore. Now, if you're one of those people that's $400 away from being bankrupt, what are you eating? Are you getting vitamins? I mean, there's got to be an impact just from that alone. You can't live on McDonald's.
The name of that filter that I have under my sink is Brondell, B as in boy, RONDELL. It's available on Amazon. It comes in either one filter or a pack of 3. I got the one that was 3. Yeah. And I think I paid $150 for it. And if you check Consumer Reports, it gets very good ratings.
Does it filter radium?
No.
I don't know. I'll let you. It gave glowing report about that.
Now, did you say radium or radon? They're two different things.
I think radium says EWG, Environmental Working Group.
I don't know. You'd have to check their website.
Patty, you're on mute if you want to say something.
Yep.
There you go.
There's another story about red meat, and that is most studies that have done on red meat and eggs and so forth are done on animals that are essentially abused. They're fed the wrong diet, their environment's terrible. So that's what they're doing the studies on. But one wonders what your health impact would be if you ate pasture-raised, humane-raised animals. I wonder.
Well, triglycerides are still there, right? It's animal fats.
Yes, but there are people who feel that saturated fat and animal fats are really not the problem for health, that it's much more the high amount of sugar that our society is eating.
Well, talk to my cardiologist about that, because he's all over me with the triglycerides.
And there are people who feel that, what your cholesterol level isn't the whole story. It's the ratios of triglyceride and
Yeah.
other breakdown, categories of fats that are important. So some people can have a high-ish cholesterol, but their ratios are very good. So then that's not so terrible. And also there are people with good cholesterol and good triglycerides who have heart attacks. So now what is that? Maybe it's not your fat levels, blood levels, but rather the inflammation that's the problem.
One of the things they found out recently is that olive oil, which I thought was a terrible food, is actually quite beneficial and is the major reason that the Mediterranean diet works, particularly what's called high phenol virgin olive oil. It always has to be extra virgin olive oil. And the Greeks have developed a magnetic resonance machine that actually can count the level of phenols in olive oil, which means that you don't have to drink or consume lots of olive oil, which is a good fat anyway. It's a monosaturated fat with about 15% saturated fat. But if you buy olive oil, which I've just recently done, you have to keep it cool and in the dark, and do not buy is not opaque, that blocks out light. If you see it in the supermarket, it says extra virgin olive oil, and it's clear, you're buying a degraded product.
There's another story about olive oil too, and that is people have found that most of the olive oil on supermarket shelves is adulterated and isn't all olive oil. So you have to be careful. It has to do with the mafia in Italy or something.
Yeah, I buy Greek olive oil.
Get it from Crete. That's the pure stuff. Crete olive oil, not Italian, none of the other. They're all adulterated. And I wanted to mention something to Arthur. When you said you don't care about the water that you shower with. My son was renting a beautiful place for a while, and they discovered that his wife and his daughter's hair was getting green. And it turns out it was because there was copper in the water. And now my daughter-in-law, who started with the green hair, has now the onset of Alzheimer's at age 60. Wow. So I think it's important to make sure that whatever water comes to our body in one form or another better be good water.
Oh my gosh. Yeah, I amend that. I take that back.
I mean, green hair, I couldn't believe it. It turned green.
Yes, and it isn't only copper. We absorb almost everything through our
right, in our water,
yeah.
Yeah, and in the shower, it's not just through the skin. You inhale a fair amount of very aerosolized water.
How much safer we'll be when we're dead? If anybody wants to know the name of the olive oil company that I have discovered, I'll gladly put that up. I actually spoke to a guy in Greece who gave me the name of the company, and they track the olive oil from the time it's picked to the time it's sent. And this is a good time to buy olive oil because it's winter and the crop for 2022 is now becoming available. And I bought 6 bottles and I have frozen 5 of them. And they can stay frozen and then unfreeze. That's another thing I learned about it. Don't leave olive oil out on the counter. You have to keep it between 55 and 60 degrees, which I can do now under a sink that's cold, but in the warm weather, I'll just keep it in the fridge and take it out when needed. or either buy a cooler. Very interesting new research about the phenols in olive oil. That's all I got. and that isn't the right spelling of my name. I'm not Arthur.
Yeah, you control that, I think, so.
I don't know. I never liked my name, so maybe I'm going to change it to Ben-Hur eventually.
Here, I'll rename you. How's that?
Please.
Yeah,
send-
but the original spelling was, I think, something that your computer generated. So you may want to try.
Yeah. Oh, there it is. Who did that? Ron?
I did that.
You did that? Thank you. Thank you. My interest in nutrition came from, I was poisoned by mercury and chemicals growing up on a summer basis. Ron knows this and Lori knows this. My grandparents had a farm in upstate New York. And in the 1950s, my father and I would drive up to Brooklyn, from Brooklyn to outside Newburgh, New York. We had a 140-acre farm and would spend the weekend spraying our apple trees starting April 20th approximately through September. And we used mercury, DDT, malathion, parathion, glutathione, Captan 7, and on occasion, before Captan came, we used mercury.
It's a good thing you're so well preserved, Art.
I've told you this before. For years, mosquitoes would not come near my father or myself. This is not a story. This is absolute fact. It exuded and it had a negative effect on my health for years and years and years, which prompted my interest in, because doctors were no help in using high levels of vitamin C and looking for, magic things that would help me that weren't in the literature. And and I was a smoker at the time. So, that fact that I'm 77 and talking to you guys is amazing.
I'm impressed. A short story. I was at my cardiologist and we were talking about my weight problem. And this was just before he was going to retire. And I made a joke. I said, well, I have a demethylated agouti gene. And he gave me a quizzical look. He says, I don't know what that means. So, sometimes you ask your doctor a question, you're expecting a really good answer and he doesn't know what you're talking about.
He thought you were talking Italian. You got a agouti gene.
Yeah, right.
Back in the day, I read a book. I can't remember the name of it, but it was before 1990. I mean, we talk about, taking the water we drink and everything. And some of these things, a laboratory rat would have to eat 8 times its body weight to be affected by this offensive material. So that I'm not sure that, my water supply is going to change my life that much.
Mm-hmm.
Well, enough copper to make your hair green is certainly different from minor trace amounts of some exotic chemical.
Well, the bigger concern is actually because, it's no longer green, but the Alzheimer's had started. And now we're worrying about my granddaughter. Maybe she'll get something as well as she gets older. Ron, as far as your cardiologist not knowing that, my first degree, and that was a long time ago, and I know nothing about it anymore, was therapeutic dietetics. In Europe, you get a degree in that. And when I came to America in 1960, and I don't think it has changed much. The only thing that the majority of MDs know about. When I mentioned the word, what I had studied and what I got a good degree in, there's famous people that I worked with, the only thing they knew about diet is something to make you lose weight. And that's when I said, I'm finished in this country. I'm not even going to use the word anymore, because that's what they consider having diet, rather than eating the right food keeps you from getting sick. We cured, after the war, we cured diabetes. Germany had no diabetes of any, you couldn't even measure it, because nobody had any sugar to eat after the war. It was the end of it. Then I met a young gentleman from Hawaii who had written a book called The Sugar-Coated Fortress. And what he told me was that we got even with the Western world. We brought sugar to you. That was it. And it's just, and we still, you're talking about all the plastics. Look at all the sugar we have in our foods. In anything, in everything, in baby food, in everything, we have sugar. We get you can refuse to eat sugar and you're still full of sugar. It's an empty calorie. It's poison, period. And it couldn't, even if it's in the form of honey, anything with the sugar molecules in it, that will do us in. Anyway.
I did an exercise at ShopRite for Karen's diet for her staff. And I went aisle after aisle after aisle, and almost every single product had sugar in it. It was incredible. Yeah.
And corn is in everything.
And salt.
And fat.
A lot of salt in
right.
Almost every food.
Yeah, the big thing now that they're talking about is fasting, which is what's old is now new. That creates what's called autophagy in the body. When the body is in a fasting state, the cells that are problematic, the senescent cells, are jettisoned. And my daughter has been on the intermittent fasting and feels much better on it. Although I read things mostly favorable, and it's an old idea. That's, it's like you were talking about in Germany and sugar. In World War II in Denmark, which in Holland, the heart attack rates went also down to almost zero, because the Nazis took all the meat. And people were not eating a high fat, high saturated fat diet. They were hardly, able to meet their caloric needs. And there was very, very little meat. The other thing that's interesting, this whole discussion about high protein, low protein, the latest, I don't know what anybody else has been reading about this, is that when you're young, you need that high protein. It really helps build the cells, builds the body. But the danger of a high protein diet when you're old, although they say to eat somewhat more when you're older, is that it helps to build cancer. And that the longest lived people are on a very moderate protein diet, because I think it's IFG1 is increased by too much protein when we're older, particularly animal protein. We're going into a different program now.
I just wanted to get back to one more thing that Art just said about fasting. The famous person that I worked with was Dr. Buchinger, who started the fasting sanitarium at Lake Constance, which people had to spend a lot of money to come to us and not eat anything at all. And they all got, they didn't just lose weight, they got healthy, But together with the fasting, we had to give them liver packs, and we had to do so many things that support the idea of fasting. What you hear now, please stay away from it, because it isn't based on anything scientific at all. Dr. Buchinger did such incredible things. He was 85 years old, and I was in my late 20s, and I had to accompany him 3 times a week, 3 or 4 times a week, hiking up this incredible mountain that had me not being able to breathe anymore. And the guy was 84, and he was healthy as anything I couldn't imagine. So whatever we did was the right thing, and it's become even more expensive. And now only the people from Dubai and stuff like that come to that Lake Constance Sanitarium to get well again. And we got rid of people with head diabetes, with nephritis, you name it. And in addition, of course, they lost a lot of weight. Hopefully they would stay with it. But during the 4 weeks that they were on fasting, we had to be very, very careful to make sure that we did all the right things that support the idea of fasting. But so I would say stay away from what they call fasting now, because it's just another one of those in things at the moment. Thank you.
I just want to make a point that despite all this doom and gloom, Americans are living longer. Maybe the quality of life has been sacrificed, but our longevity is increasing.
Actually, they're living less than other developed countries because of our opioid crisis. But that's a different thing. It's not, you know, if you stay away from the opioid, you're right. Most people can live longer.
Anybody here on opioids? Anybody? No, I think we're okay.
But it would be interesting to hear more research being done to attack these addictions through the epigenetic treatments.
Yeah, I think you're going to hear more and more about epigenetics going forward, because unfortunately, when they advertise a new treatment on television and they talk about a drug or whatever, they give you some made-up name. They don't tell you how they came up with it. They don't tell you how it works. Maybe some of it is epigenetic. We just don't know, because you know they don't treat us like we're intelligent. So we're not. We're not.
But
educated.
I would just be concerned that in trying to treat that, they cause all my liver cells to think that they're lung cells, and then I'm in trouble.
That would be bad. That would be bad. One of the interesting things that they figured out about epigenetics was that if you can go in and you can reset all of the tagging, you end up with stem cells, right? That's how they can take skin cells and turn them into stem cells again.
So I'm going to say goodbye, everyone.
Bye-bye.
Yeah, but there's very interesting stuff.
Yeah. Yeah, and nobody knows about it. It's one of the great unknowns today of what's going on in science. And you know, it's really shocking to me, and this is the last I'm going to say about any of it. I was doing a talk in AltSpace about the scientific method. And when I did the research in the scientific method that we do today, what we call the scientific method, is 80 years old. We've only had the scientific method as we know it for a very short time. DNA was discovered in 1944. This stuff is so new. You know, I mean, yeah, 44 was before I was born. So yeah, it's a long time ago. But if, you know, in the history of humanity, it's like yesterday.
Yeah.
You know,
Wasn't
and
it Crick and Watson who got the
They figured out the structure of it,
Yeah,
atomic structure,
spiral.
but the notion that it existed was earlier.
was 8 years earlier or so, or 10 years earlier.
Yeah, 44. But I mean, that's how new everything is, you know, and epigenetics wasn't even considered as a possibility until the 70s, and it wasn't formalized until the 90s. I mean, that's not that long ago, 30 years ago. So we've got a lot to learn, but I imagine we're going to learn it quickly, because now we have the scientific method and we do know that there's epigenetics, and maybe they'll figure out what the other 40% of our DNA is actually doing. Hang in there.
I think you said to me one time, Ron, that if you can stay healthy, if you're young now, if you're born now, or you're 10 or 15 or 20 years old, your chances, assuming the world doesn't, implode, of living, into the past 100 in a healthy state are getting better all the time.
Oh yeah. Yeah, if you can live to be 100, you might live to be 200. And if you can live to be 200,
Yeah.
who knows? You know?
Yeah. Social Security. Well.
Okay, listen, I'm hungry.
Yeah.
Yeah.
All this talk about food. I'll say goodbye to everybody. Nice seeing you. Thanks, Ron.
Yeah. Thank you all for coming.
Thank you.
Bye-bye.
Thank you. Bye.